All right. Thank you, everyone who is joining. My name is Lenny Teytelman. I'm one of the co-founders of Protocols.io. I'm a scientist by training, PhD in genetics and genomics, both experimental wet lab work and computational biology. And I co-founded Protocols.io based on my postdoc experience. So it was actually my troubles and troubleshooting as a postdoc at MIT after I got my PhD, where I spent a year and a half correcting just one step of the single cell microscopy, RNA microscopy protocol in yeast. And instead of a microliter of an enzyme in one particular step, I needed 15. And instead of a short incubation for 5 minutes, it turned out needed an hour. And after a year and a half of figuring out why the protocol wasn't working in my hands and correcting it, I realized that it's not a new technique. So I don't get credit for it because it's not a new paper. It's a correction of something previously published. And everyone else who's using this protocol is either getting completely misleading results or has to spend a year or more, like I did, figuring out the correction. So that experience was the motivation behind the obsession with creating a central place where we can exchange this knowledge. So that's the story behind Protocols.io. We got started in 2014. I'll say a little bit more about our history shortly, but let me share the screen and just do sort of housekeeping information. So as I mentioned, this is being recorded, and we'll share the link to the slides that I'm showing and the live demo part of Protocols.io. And this video we'll send to everyone who registered. So if there are any URLs or anything like that, you don't need to scribble them down. You'll get tomorrow or sometime later today the email with the link to the slides, all of the URLs, and the video recording. I'll leave plenty of room at the end of the webinar for Q&A. We'll go about 45 minutes of me presenting, and then we'll have time for Q&A. And obviously, I won't go over all of the functionality and all of the amazing things that Protocols.io can do. That would take a lot longer than an hour. So this is an introductory webinar, assuming that you essentially are new to the platform and haven't used it much before. If you're a power user, if you already have protocols and you're about to share them publicly, we have other webinars that we offer every month, so you can check them out at this link. There is an advanced webinar, there is a getting credit and recognition if you're in academia. So you can see what are the other webinars that we offer, and maybe now an introductory webinar is helpful, but maybe next month a more advanced one is something that you can benefit from. So that's the link to the webinars. And just like the intro one, we offer them in different time zones. So you should be able to always find the webinar that you want this or next month in a time zone that works for you. And if there are any questions as you use Protocols.io, you can always email us. There is a help button on every page of Protocols.io. So we do provide excellent customer support. There are several scientists like me on the team. We alternate the webinars. We alternate answering questions. But also, as questions pop up, as you use the platform, you don't have to wait a month for a webinar, advanced webinar, to figure something out. There is a link for one-on-one demos. You pick a time for the week that works for you and schedule a 30-minute session, and one of us will jump on a Zoom call and help try to share the screen and help to answer the questions that you have. And we also have on this help link in general, we have a very extensive help section So anything we don't cover, most of the things that you have questions about should be on the help page. So please don't hesitate to take advantage of all of these resources. Right, and now let's jump in. So we'll start with a little bit of motivation, not just my personal story of why I became obsessed with creating protocols IO, but we'll start in general about the importance of sharing methods. Then a summary of the key functionality and the mission of protocols IO, and then we'll dive a little bit in a live demo of the key parts in terms of using the protocols, the key functionality we'll cover in here, and just getting started, creating, sharing protocols, publishing them if you are an academic rather than biopharma. And Protocols IO serves both academic and industry users. But obviously, if you're academic, the publishing part is more relevant for you. If you're in a pharmaceutical, you'll be using it in private mode and sharing across your company rather than making things public and getting credit for the methods. And as we go along, if you have questions, feel free to add them into the Q&A in the Zoom control that you should see at the bottom if you mouse over. All right. So some of my favorite, other than my personal story, some of my favorite tweets about the challenges of finding protocols and repeating works of others are highlighted here. I have a long collection on where I've bookmarked Twitter stories like this, but here's one from a postdoc at University of California, Riverside, who says, I'm looking for a protocol in a 1997 paper, as described in 96, finds 96 paper, as described in 87. It's like peeling layers of an onion. Finds 87 paper, and even University of California doesn't have subscription to it. It's a common and frustrating experience. This is from a biologist. This is from a physicist looking at a method section, and it's, again, like layers of an onion. Devices were fabricated as previously described, and the original reference just says devices were fabricated with conventional methods. And so it is really hard from the method sections of papers to figure out exactly what was done and build on the work. We have to contact the individual labs, ask for detailed protocols, or search around hoping that someone else knows how to do a particular technique. And there was in biology a systematic effort to look at how easy is it to reproduce published literature in cancer biology, where Center for Open Science and Science Exchange, two organizations, did a study that was, I think, like $1.5 million over several years, where they picked 50 papers from high-profile journals in molecular biology on cancer, and just tried to repeat independently key results from those papers. And there was a lot of media attention to the results of this work. And writing in The Atlantic, science journalist Ed Young, part of his coverage of this work was that the hardest part by far was figuring out exactly what the original labs actually did. Scientific papers come with method sections that theoretically ought to provide recipes for doing the same experiments, but often those recipes are incomplete, missing out important steps, details, or ingredients. In some cases, the recipes are not described at all. Researchers simply cite an earlier study that used a similar technique. So this obviously is in line with what we saw in the individual tweets. But then after several years, the whole initiative wound down at just 18 out of 50 papers. And on the summary page at Center for Open Science, when they talk about this work, they say that 0% of the papers had fully described protocols accompanying them. And of course, the method sections don't have the full information. But importantly, in this initiative, the scientists who were trying to do the replication, they actually contacted the laboratories that did the work. They contacted the groups. And even when collaborating with the scientists with the authors who published the work, they often could not find the actual protocol. So it's not just that the papers we publish in the journals don't have enough information. The problem goes deeper inside our own labs. We're not organized enough and they're not tracking in a good enough manner what we're doing so that when someone leaves and a new person joins, it's easy to see exactly what was done for which experiment and to build on those results. So this was quite eye-opening, and this is exactly what Protocols.io tries to improve. So if we talk about our mission, it is to make it easy to share the method details before, during, and after publication. And the before and during the work, and as you're preparing to publish, these are really important. So Protocols.io is not just a public repository, but it is a tool for securely organizing and collaborating on protocol development in teams, whether you're in academia or industry. And as I said, we launched in 2014. So as a snapshot, at this point, there are over 160,000 registered users, researchers who are on the platform. You can see that there's over 16,000 public protocols that these researchers have shared, and about more than 67,000 that are in the private mode. And just to be transparent, if you're using Protocols.io for public sharing, if you're academic and you're publishing, the protocols are open access. They are free to read, free to publish. So the public sharing is free for the authors, it's free for the readers, and our business model, kind of like GitHub, is if you're sharing publicly, it's free. If you're using Protocols.io for private collaboration to organize the protocols within your lab at a university or in your company, that's the part that we charge for. And for many academics, actually, Protocols.io is free even for private use if your university pays for an institutional license. So you can pay directly for your own workspace or for a lab if you're academic. But if you encourage your institution, if you don't already have a site license, if you encourage your institution to sign up, then it becomes free for private use as well. And here we just highlight that unlike a traditional journal, which focuses on sharing your work and helping people discover protocols that they need, Protocols.io, as I'll show in the live demo, is really embedded in all parts of the research workflow. So as you're designing, as you're refining your methods, as you're doing the work, Protocols.io is a tool for all of those parts. And then if you're academic, if and when you're ready, it helps you to share your protocol and get credit for it. Right. So long before publishing, you can easily share. There is collaborative editing, concurrent editing, just as on Google Docs, with a record of who made what changes. So you can evolve the protocols and refine them, get feedback from colleagues. And as you're performing the experiments, I'll show what the run functionality is, but there is a really cool feature of Protocols.io that helps you to run the protocols as you perform the work, record what you've done. And if you're academic, at the end, it is very easy to get credit for your method development, to share your protocols, and it helps you to, as I'll show in a second, it helps you to publish your research papers and to detail your protocol so that we avoid, in the method sections, we avoid the scenarios that we discussed just above. And here's an example of how Protocols.io helps you write method sections. There are many journals that in author guidelines encourage use of Protocols.io. And so in this example paper in PLOS Biology, if you go to the materials and methods, you can see it says methods and protocols for the following manipulations and preparation are available as a collection in Protocols.io. And I think it's a single DOI, it's a single link. And if you click on it, it takes you to the collection on Protocols.io with 10 different buffers, media preparation, and detailed protocols that are part of the work, right? On your own protocols, in addition to you making a protocol public as part of a research paper, you actually, again, if you're academic, you can send your protocol for peer review and turn it into a peer-reviewed publication in one of these 3 journals. So we have partnerships. Obviously, you can send them to a journal of choice, but outside of these ones, but these are protocol publishing journals that specifically welcome and peer review protocols, even if you don't have new and exciting results. But if you've spent half a year refining a method and getting something that works on liver tissue to work on heart cells, or something that works in grasshoppers to work on crickets, if there is substantial work that has gone into optimizing and tweaking protocols, you can easily click submit for peer review and turn the protocol that you've shared on Protocols.io into a peer-reviewed paper. And this is an example from one of these 3 journals, and PLOS One, that's the publisher with which we've had the longest running partnership towards this, but there are now many examples of such papers where the protocol points to the peer-reviewed paper and the peer-reviewed paper points to the detailed protocol on Protocols.io. And in terms of the public repository, one of my favorite examples is a discussion on Twitter where a postdoc is saying that I'm looking for a method to do RNA extraction in primary cortical neuron cultures. The details of the science are not important. And a postdoc from UCSF says, from the ones on Protocols.io, here's the one that should work for you. And what I love about this example is that when you go to the actual paper that this protocol is part of, it's a paper on 3-spine stickleback parasites, right? Fish parasites. And the authors, it's actually a group from Australia that published this, but the authors, not only did they make their paper more reproducible by sharing the protocol, detailed protocol as part of the method section, but by putting it in our repository, they made it available to everyone. And, the chances that someone who's looking for protocol and cortical neuron cultures would be familiar with this paper and find the protocol are very low. But once you share it in a repository, just like sharing protein or DNA sequences in a central database, in a central repository, it becomes something that can be used in other applications. And so it enhances reuse and adoption beyond the immediate audience of this particular paper. And with that, I'm going to actually stop the slides and go into a short demo of some of the key functionality of Protocols.io. So this is the landing page on Protocols.io. You don't need to register or sign in to search and take a look at those 16,000 plus public protocols that I mentioned. But of course, if you create an account, then a lot of functionality opens up to you rather than just being able to view a protocol or print it. So I'll highlight some of those benefits here, but as I said in the beginning, obviously not everything. But the most important thing to note when you're looking at a protocol is that this is not just a PDF or static text. The protocols on Protocols.io are dynamic and interactive. So you'll notice that if I mouse over any step, I can actually ask a question that will go to the authors, that will go to everyone who's using the protocol. I can ask a question publicly or privately, depending on my preference. And then the responses actually you can see building up. So here's a question on step 2 and answer from the author, and you can see threads, you can see the discussion sort of building up like an FAQ document right on the steps. So instead of answering a question by email or in Slack, and then you get it again, and you have to find that email or answer the question again, you build up the FAQ right on the protocol itself instead of having to copy yourself. And eventually, people, if they're looking at the protocol, will notice the discussions and hopefully stop asking you that question because the answer is right there. And if you're getting questions and you want to clarify your protocol, even after you've published it, it's really easy to click create new version. So you can see this protocol is on version 2, and you can easily create a new version and share corrections and optimizations. You can clarify things based on those discussions that are happening. And then under the More button, between the versions, there is actually a compare button that allows you to see in the original and in version 2 which steps are the same, which ones are different. So it'll highlight for you the identical steps. You can see 3 through 8 are the same, and then something changed in step 9, step 10, and then 12, 13, 14 are the same again. So it's easy to version. The previous versions don't disappear, and you don't have to strive for perfection, but when you're ready, you can share things. You can say this is a protocol that is a work in progress as you share it, and then you can share version 2, 3 to improve it, and then say this one is the final version that we're using. But even when you share and say it's final, you can still share corrections and optimizations. And if someone comes to a link to a version that was shared previously, we will let them know that there is a newer version that is available. So if someone lands on version 1, there will be a pop-up that tells them you can see version 1, but there is a newer one available. So we'll guide everyone to the more recent one automatically. And then you'll also notice, this is really important, you'll also notice that every protocol, public or private, has this copy fork button. So essentially, every protocol serves as a template. So if you're working with corn and I'm working with Arabidopsis, I can take your protocol, create a copy of it, and depending on the organism species that I'm working with, depending on the equipment, and in my research team, I can make a copy of your protocol, and then it becomes editable, and I can make edits and changes to it. And if and when I'm ready, then I can share exactly what my fork is. I can make it public, and we give credit to the original authors. We'll indicate clearly that this protocol is a fork, but we'll give credit to you for sharing the optimizations for Arabidopsis relative to corn. And so both the original authors and you can get credit for it. And that compare functionality that I showed also applies not just to versions, but to the forks that people create. And in addition to when I said that the protocols are dynamic and interactive, I also wanted to mention that not only can you mouse over a step and ask a question, but we have a really rich editor for creating protocols that's specifically designed for capturing the details that you would need to replicate and build on a technique for reproducibility. It's very easy to add reagents that you've used. We have catalogs of many reagent vendors, so you can just put in a catalog number and say exactly where you ordered and what chemical you've used. If it's a computational protocol, you can share which data set, which version of a library, which script you use, link to GitHub. It's very easy to add not just images, but videos. And we're starting to see more and more protocols that have videos in them. And that, of course, helps a lot, especially if someone who's reading a protocol is not a native English speaker. Creating videos is really helpful for localization and conveying the nuance of the protocol. So you can take your phone into the field, into a cold room, shoot a 15-second or 1-minute video, add it right into the step, and it helps tremendously. It's like someone coming to your university to see and watch how exactly you perform a technique. And the last thing that I wanted to show on the protocol level is this run functionality. And this part is particularly helpful for experimental wet lab or field methods that are hands-on. And I think out of the 16,000, 17,000 protocols, public protocols that we have, I think about 5 to 10% are computational, but the majority are hands-on protocols. And for those, this run button is really nice. So this allows you, when you're performing the experiment, to actually step through it, right? So it's kind of like a checklist app. I can run it on my iPad, on a Samsung tablet. And as I'm doing the work, I can say, I can see where I am, what I'm doing next. I'm done with step 3. It keeps a record of when you finished it, right? There's a timestamp. And if I change anything as I'm doing the work, I can click edit and I can say, today I did it not at room temperature, but at 30 degrees Celsius. And if I save it, I have a record of exactly how I ran this procedure on that particular day. And then I can export it to my lab notebook or just have it in protocols.io and can come back to see what I've modified and how it deviated from the protocol itself. So this is a really nice feature. And then if we exit out of the protocol view, if you have an account and you sign into protocols.io, I wanted to show you what the file manager space looks like. So here I am signed in, and this is the view that you'll have. It's kind of like a file manager on your own computer. We call these workspaces. So you can have one or more workspaces, which is not, it can be your individual, but you can have collaborators. Some workspaces have 10 members, some have hundreds of members, but you can create a structure with folders and subfolders. You can organize your protocols. And if this is a workspace that's shared across a company or for an entire research lab, all of the members, as soon as you add a protocol, have access to it, can use it, can run it, can comment on it. So it becomes an organizing and collaborative tool, not just a publishing tool in that sense. And I also want to highlight, if you're new to protocols.io, I want to highlight that it's really easy to get existing protocols into protocols.io. There are a few ways of doing it. So if you click new, create a new protocol, it's going to be empty. Here's an example that I've created. Let's delete the steps that are here. So this is a new protocol. It's organized at the top into different sections. There are the step-by-step instructions for the hands-on work. There is background information, warnings, guidelines that you want to show up, materials that you're using. The description should include the abstract of the protocol, authors, keywords that would help to find it in the workspace or if you make it public. And then for the steps, I wanted to say that, of course, you can create steps one by one. But if you have an existing protocol, you'll notice that there's also the option to insert steps from a text file. So if you have a PDF or an existing Word or Google document, you can very quickly get that into Protocols.io. So here's an existing protocol in Google Doc. I'm going to copy the steps out of it, paste them here, and each one is numbered. So I'm going to choose option number 1. Each number is a step. And then instead of adding these one by one, I've quickly created the protocol. You don't need to click save, it auto saves. And here it is. And then you can refine it, add components, add a video like we talked about, add sections to create a table of contents. So you can do a lot of work to make it better, but for existing protocols, it's quite easy to get the relevant information in fairly quickly. This takes a couple of minutes, but obviously takes advantage of Protocols.io. If you don't have a couple of minutes, if you have 30 existing protocols as PDFs, I wanted to show some of the other options. So instead of creating individual protocols, if we go back to the workspace, you'll notice that not only can I have protocols, which are these items in here, but you'll see that they're actually PDF files. So the easiest thing you could do is take Excel, PDF, Word files, and just drag and drop them into the workspace, into the right folder. And even though you don't have a DOI and you can't really publish it that way, but you've instantly organized them in a collaborative space. But essentially, it's then kind of like Dropbox. So you could take all of your companies or teams protocols, 30, 100 of them, just drag and drop them into the right space. And it doesn't take full advantage of Protocols.io, but in the very least, you've now shared them across the entire team, and they're in a central cloud organized space. But if you have a few more seconds, what I want to highlight, my recommendation in between adding step-by-step protocols and just dropping protocols in as a PDF, one thing you can do is create a new protocol, not fill out the steps, but just add title for it, add an abstract that'll help you to remember what this protocol is. And then if you scroll down on the description tab, you can add attachments to the protocol. So open up your file browser, add the PDF attachment, and then you've created a protocol shell. The PDF is there, but this way, instead of it just being a file, it's now much easier to find as you search through the keywords, the abstract, the title are more discoverable. So this will take you, 15 seconds per protocol that exists to get it in. Ideally, of course, if you really want to take advantage of Protocols.io functionality and the run functionality and the many features that I won't have time to go over on this webinar, then ideally you do spend a little bit more time and enter step-by-step instructions. Those of you that are from companies or universities that have a subscription to Protocols.io, you actually have another option for getting your protocols in. And let me spend just a minute showing that. So if you go to Protocols.io for institutions, there's a link in the slides to it. You can actually, if you're from an academic institution, If you scroll down, there's an option to see if your university has a subscription to Protocols.io. And if it does, you can get more information here. But if you are on a premium plan from your university or company, that actually includes protocol import assisted by our editorial team. So in that case, you can take an existing protocol. You go to, we enter protocols. Again, the link is in the slides. You just drop the file in here. It'll go to one of our editors who will enter it. Another editor with science background. All of our editors have science background, but one editor will enter it. Another one will review it to make sure that there are no mistakes, and then privately reassign it to you, share it with you for review. You give us feedback if you want anything changed, and then the protocol gets, when you're happy with it, gets reassigned to you. So if you're an institutional plan, that's something that's available. If you're not on an institutional plan, for simple protocols, it's $50, $75 just to cover work, but that's another option. If you don't want to enter the protocols yourself, you can take advantage of our editorial support for this. And again, if you're part of a company or a university that has a site license, then you don't have to pay for this import. It's included with the site licenses. Right. So these are the key things that I wanted to go over. As I said, we'll share the recording, and the slides have a lot of these links and information. I just went over most of the stuff. I didn't go over the editor in detail, but we don't have time for everything. I showed how to import existing protocols. This is a review of the different options that are available for getting started, including the import service, some tips on creating good protocols, and I think we've gone over most of this. Also, for those of you who are in academic institutions, if you have a site license, Protocols.io is really handy in lab classes or computational classes where you want to share hands-on instructions with the students. So if you have an undergraduate lab class, so there's a lot of use of Protocols.io for teaching as well. And as I mentioned, there are hundreds of journals that have Protocols.io and author guidelines. There are more and more funders that recommend or require sharing protocols and recommend Protocols.io specifically, and more universities that have site licenses. So take a look to see if you're covered by it. And with that, I think we are exactly 40 minutes in. And so I will stop there and see if there are any questions. You can, if you raise your hand, you have a raise hand button on your screen. If you raise your hand, I can also unmute you. We're not a giant group of hundreds of people, so if you have questions, I can unmute you, or you can put them into the chat or the Q&A window. And if you don't have questions, then have fun exploring the features, and don't forget that we always answer questions quickly, and we have other webinars, like I said in the beginning. And you can always jump on a one-on-one demo if there's something that we didn't cover. Or if you don't have a question now, but have a question later, we are available to answer them. So thank you everyone for joining, and I'll hang around. I'll stop recording now, and I'll hang around a little bit longer to see if anyone has questions.